I will briefly introduce myself. I'm a researcher at the Trieste outstation of the Institute of Crystallography, and I graduated in biology at the University of Trieste. I did my PhD at SISA on the structure and functional characterization of two antibodies with therapeutic interest. And then I worked for 10 years, more or less, developing therapeutic antibodies, working first at Line Genomics and then at Rotafarm Biotech, where I was the leader of the protein science group. And we had to work cloning, expressing, purifying, and in vitro characterizing by functional studies our leads. Then I moved back to academia and I joined the institute where I'm working now. Briefly, I recap our expertise. We are interested in the structural and functional characterization of therapeutic targets and protein involved in disease. And with the aim to develop new effective drugs and to find out the molecular determinants underlying human pathologies. We usually do that by structural characterization, so by X-ray crystallography and biosacs mainly, but recently Francesca joined us and she's an expert of NMR, and hopefully all of us will be experts also in cryo-EM with this project. And then regarding the functional characterization is performed by biochemical and biophysical assays like CD, fluorescence, DSF, DLS. And the in vitro functional characterization is the topic of my talk, because it's done with the core of our facility that is based on GCI and ITC and will be improved thanks to this PRP CERIC project. I will start with the isothermal titration calorimetry. This is a very powerful technique that is based on a very simple principle, is the fact that each interaction provokes an exchange of heat. Or a release of heat, or the heat is absorbed upon binding. And so measuring the heat that is exchanged, this technique can give you a lot of information, not only the binding affinity, that is what we usually can measure with a lot of techniques, but also the stoichiometry, that is not trivial from many other techniques, and more importantly, the thermodynamic profile. This is the only technique that gives you information about the enthalpy that is, and so the entropy and the free Gibbs energy that is linked to the interaction. This is an important parameter, an important characterization, because as you can see in this graph here, can you see the pointer? No? Okay. As you see in this graph, all these three interactions seem to be the same because they have the same KD. As you know, the KD is linked by a mathematical formula to the free energy variation. But the mechanism of the interaction are completely different. The first one is enthalpically driven. So it's due to the formation of hydrogen bonds, salt bridges, van der Waals interaction. In the second case, the interaction is entropically driven, And so it is due to the formation of hydrophobic interaction. The third one has a positive contribution of both entropy and enthalpy. This characterization, so ITC studies are very important in the different steps of the pipeline of development of a drug, because the first step is after the high throughput screening that can be performed by DSF or also by GCI. I will talk about this high throughput technique later, or SPR, for example. The lead that can be selected in this way can be first validated by ITC, and then by structure-based drug design, the lead optimization can be obtained by means also of ITC information. How does this system work? The experiment we perform is based on this very old microcalorimeter. We are going to upgrade. And the microcalorimeter is characterized by a thermal core that contains two cells, a sample cell and a reference cell. In the sample cell, you put around 0.3 milliliter of the sample at a concentration, a starting concentration between 10 and 20 micromolar. The concentration has to be optimized because it depends on the amount of the heat that is exchanged. And this is a characteristic of each interaction. Then you fill up the syringe with the so-called ligand that is tenfold concentrated respect to the sample. You load more or less 40 microliters of the ligand, and the experiment consists in a series of injection of the ligand inside the sample cell. We inject a small volume from 0.5 to 1 microliter till we get the saturation of the system. And it depends on the stoichiometry of the interaction, the excess of ligand you have to add to the sample. At each injection results in an exchange of heat that can be measured, and then after integrating the integration of each peak to calculate the total amount of heat that is exchanged, we can plot the integrated heat in function of the molar ratio. That is the ratio between the concentration of the ligand and the sample. And in this way, you get the so-called binding isotherm, and the slope allows you to calculate the KD. The midpoint of the transition of this sigmoid curve gives you the stoichiometry, and the height between the beginning and the end of the transition gives you the enthalpy contribution, and so allows you to characterize the thermodynamic profile of the interaction. I will present you three case of studies, experiment we performed in our facility, and unfortunately they are all about the interaction between proteins and small molecules, even if there are apparently no limitation in molecular weight or in the molecular weight of the interactors. This is let's say an internal project, is something we are working with Francesca for several years, and is about the characterization of the interaction between ATP and the NGF pro-NGF system. These two proteins are known to exert opposite function in the development and maintenance of the nervous system. To characterize the interaction that Francesca also studied by means of NMR, we put the NGF at 40 micromolar concentration in the cell, and in the syringe ATP at 5 millimolar concentration. We perform 40 injections of 1 microliter, saturating the system, and we could get an affinity that is quite low. Like the KD is around 1 millimolar, and as you can see, the interaction is enthalpically driven. The interaction and the role in the modulation of pro-NGF function is completely different, as we could see by ITC characterization. We put the protein at 20 micromolar concentration in the cell. We titrated with ITP at a concentration of 2 millimolar, and by around 20 injection, we could get the saturation and the KD that is much better than the one we registered for the mature form, that is around 40 micromolar. Also, in this case, the interaction is enthalpically driven. Another interesting system where we had to change up, to switch the setup, was a project we followed in collaboration with Marta and Paola, and also Graziano from the University of Trento. We wanted to see if the two sites that were occupied by a cyclodextrin molecule in this crystal structure of PTG, that is a protein involved in glycogen metabolism, were all physiologic ones. So, but in this case, we didn't want to have high concentration of cyclodextrin because it could form micelles that could complicate a lot the interpretation of the experiment. So, fortunately, the sample of the protein sample Marta prepared was a very good one, very soluble. We could reach, she could reach a concentration of 100 micromolar. We could load on the syringe. In the cell, we put cyclodextrin at a concentration of 3 nanomolar, and by 40 injection, we could, of 1 microliter, we could get saturation and see that only one of the sites was a physiological one, with a KD of around 60 micromolar for an interaction that is mainly enthalpically driven. The last case I will show you is about a collaboration we did with the University of Padova and Ca' Foscari University of Venice. They were interested in characterizing the interaction with the perfluorooctanoic acid, PFOA. As you know, is a man-made toxic compound that is widely found in industrial products, and unfortunately highly resistant to degradation, and this is why it's a global concern for the risk to human health and the impact on the environment, especially because FOA is absorbed by our skin, and serum proteins like human serum albumin are able to veiculate to the different tissue this toxic compound. So from the crystal structure, we could see 4 binding sites, and we performed an ITC experiment to validate that all these sites were physiologic sites. We put the HSA at 120 micromolar in the cell, and we titrated with FOA at 4 millimolar concentration, and after 20 injection of 1 microliter, we could get this the saturation. And from the analysis of the binding isotherm, we could see that the best fitting was obtained postulating the presence of one high affinity site with high affinity of 0.3 on a KD of 0.3 micromolar, and 3 low affinity sites with KD that is 2 order of magnitudes higher than the previous one. And as you can see, all these interactions are both enthalpically and entropically driven. This is just to show some example of how ITC can be applied to study protein interaction with the small molecules, but also with proteins, nucleic acid, and so on. To summarize, all the advantages of this technique is, I didn't stress this point, but it is the most reliable and most native technique to study intermolecular interaction, but because it happens in solution, completely in solution, without immobilization, without any labeling, and this makes the assay quite easy to optimize. It has quite broad dynamic range of affinities. In a direct experiment, we can go from around, let's say, micromolar to nanomolar affinity, but with concentration, with the competition experiments, we can go down to picomolar and up to submillimolar. As I told you, it's very information rich. We have a lot of information about affinity, stoichiometry, and the complete thermodynamic profile, and a broad range of application because it's compatible with a wide variety of solvent and buffer condition. So till, let's say, 5 years ago, this was the main technique that we had in our hands for a quantitative characterization of intermolecular interactions. And so, but as you can see, this is a very low throughput technique, because you can do at maximum 8 experiments a day, 8 systems a day, and moreover it needs a very high amount of the sample, and you not always you have so large amount of sample to perform your measurements. And so we had to integrate this by the time this powerful technique, but very demanding, with let's say semi-quantitative and qualitative technique like gel filtration, pull down, co-IP, differential scaling fluorimetry, dichroism, circular dichroism, or fluorescence. Then we had the opportunity to complement our system by an orthogonal instrument based on GCI that could solve all the problem of isothermal titration calorimetry. And this was an opportunity in a project of Area Science Park Open Lab 2. And when we got that budget, I didn't tell you, but I was quite expert in the SPR system, in the BIACOR system. I worked with different models of BIACOR, but unfortunately our budget didn't allow us to buy this system. It was too expensive. So we look at the technology present on the market 5 years ago. And at first, we were very impressed by the BLI system from the presentation of the vendors. I have to stress this point, because this system is very user-friendly. It's very robust. Everyone can put the experiment with no particular training. It was the less expensive instrument. It needed no maintenance, and also the consumables cost was very, very, very low. But when we test this system in a demo at in our lab on our sample, realized that the performance to characterize interaction between protein and small molecules, that is a core part of our research, was very, very bad. We could not have data at all on different systems. So we evaluated different technology, and in particular, we spent a week in Wadesville at Creoptics, performing the same experiments we did with the BLI system on the wave system. And it was really another world. This is because we could get data of very high quality, and we were impressed about the quality of this data, also characterizing system interaction between protein and small molecules. So we decided to buy this system, even if the technology, it was not so young, but it was not so spread in the market, and it was a little bit more expensive in terms of maintenance and chips. Anyway, this system, beside the high data quality we could see immediately, it's also a quite robust system, and this is due to the fact that they worked a lot on the improvement of sensor technology and microfluidic system. Just to give you an idea, comparing the SPR and GCI, in the Beaker, you get smaller signal because the laser hits the chip only locally. You giving rise to a localized evanescent fields, that is the part of the chip that is able to measure differences in mass. Instead, the waveguide interferometry is characterized by a laser that travels across the sample, and in this way generates a field that gives a signal that is the result of the interaction that are happening on all the surface. Moreover, the grating couple interferometry setup allows also to reduce bulk effect and so to increase the sensitivity. Besides this, the system is is more robust compared to SPR, because they were able to integrate microfluidics inside the chips. In this way, this seems trivial, but when you worked with the Beaker, and the fluidic is part of the system of the instrument, and you clog the system, you need to spend a lot of money to exchange all the cartridge of the microfluidic cartridge, and you have to wait for the technician to come and perform everything. And And in this way, if you clog the chip, you simply throw away the chip. Okay, it's 500 euros. It's not so cheap, but it's less than 10,000 euros. That I don't know if the price is the same, but let's say 15 years ago, the price was this one. The fact that the microfluidic is integrated has also other advantages besides the robustness of the instrument, because you can have a fresh and clean microfluidics for each experiment, and you protect the sensor surface. And moreover, this setup allowed them to exclude the system of microvalves, and this makes the system much more faster and allows you to measure also very fast transition times around 0.11 second. So how does... Eccolo, vedi? How does it work? We have to capture one of the interaction partner on the surface. This is called the ligand. It can be done in a reversible way by a capture. For example, using stack proteins, or you can covalently couple the proteins by amine coupling, for example, on the surface. And then the surface of the chip is gold that is functionalized with an inert polymer. And the experiment consists in the dilution, in the injection of increasing concentration of the analyte, that is the second binding partner, to get the so-called sensograms. And from the dissociation part, you can estimate the K off, so the kinetic dissociation constant, and from the different concentration together with the value of the K off, you can get the K on, and so you can obtain the KD. And in this way, this system allows you to characterize the interaction not only by means of affinity, but also the kinetic itself of the process. Here I will go very, very fast, just to show you that it is really very true that we could address the interaction between a broad range of molecular weight. You can see here the interaction we measured between a GPCR and a small molecule. This GPCR was obtained in virus-like particles that we captured by with germ agglutinin. And this 3 megadalton ligand we captured on the surface could be measured with an analyte of less 1 kilodalton, obtaining these very, very nice sensograms. You can see the low ratio of signal to a very, very good signal to noise ratio, because the response is very, very low. And in this way, we could obtain the data that were already published, because this work was done in collaboration with Rota from Biotech to validate the sensibility of this system. Another, going very quickly, this experiment was performed with Fabiola Penta in collaboration with the University of Nova Gorica. He was studying the interaction between a tumor suppressor, one of the largest cytoplasm nucleoproteins. In this case, we did the opposite. So we captured the small protein that has an histidine tag on the surface, and we used MVP as an analyte, and we could get nice data whose specificity were confirmed also using a mutant, a point mutant that was designed to disrupt this interaction. In this case, the KD are not the results only of affinity, but also avidity, because it's the MVP protein is composed of 77 identical subunits. And so you cannot only measure the affinity, but also the rebinding. And this is why we perform also steady state analysis of this system, obtaining an affinity that is much more lower, but anyway very different from the one of the mutants. This experiment were part of the project of Paola and Marta in Graziano, and we could characterize the interaction between two proteins that are involved in glycogen metabolism, obtaining very nice curves and an affinity around 90 micromolar, and we also could characterize the interaction between the part of one of the two proteins that was directly involved in the interaction with two peptides, a longer and a smaller, and we could see that the smaller that occupies a higher surface on a binding surface at a KD that is twofold better than the short peptide. This is another system in which we measure the interaction between peptides in collaboration with our colleagues in the Catania outstation that are developing so-called beta breakers that are based on the second self-recognition motif that prevents the self-association of A beta, soluble forms of A beta that are known to be linked to the onset of Alzheimer's disease. This alpha breaker was also conjugated with porphyrin to obtain biosensor, and so we immobilized the A beta peptide and could characterize the interaction of all the peptide alpha breakers, and we could see that the interaction is specific using as a negative control the porphyrin, because the KD is 45 less in the case of the peptide, and we could see also the importance of the ion that is inside the porphyrynic ring, analyzing different conjugates, porphyrynic conjugates. Very quickly to internal projects. This was about the characterization of the interaction between a target for anti-cancer therapy, that is GLI1, that is the final effector of the HOG pathway. In collaboration with the group of Professor Botta in the University of Sapienza, we tested GLAB-B that was selected from a natural compound library, and two derivatives of this compound. And we could find that immobilizing our protein, we could find that the KD of the GLAB-B is improved in one of the derivative, but it decreases with the second one, and this was confirmed by our docking studies. A similar project was about a target. We are working in to develop antimicrobial drugs that should not give antibiotic resistance, using as a target the salicylate synthetase that is involved in the iron acquisition routes. This project, at first we tested the scaffold, a series of furan-based derivatives that have been selected as effective inhibitors of the Mycobacterium tuberculosis homologue of SAS, and we can get very nice curves, affinity in the range of from 6 to 60 micromolar that could be rationalized by docking experiments. So to switch to the other advantage of GCI is the fact that you can also deal with a very high number of analytes, so you can perform high throughput screening. We perform a nice throughput screening against SAS using a fragment library of 100 compounds, and we could select a series of compounds that we have ranked considering the response. Well, the best performing in this screening, we recently crystallized, and we could see that the binding happens at the active site of the protein. And so now we are working co-crystallizing the other compound fragment we have selected. Very quickly, this is a project in collaboration with the University of Trieste, a screening for for the development of anti-cancer drugs against two transcription factor, HMGA and 1 and 2. Luca Braga give us a library of kindly give us a library of 1,300 around compounds. We were able to screen in 3 days against on plates of 384 wells, and obtaining a series of positive that some of them were specific for one protein, some others to the other one, and in the end we decided to focus on the one that were effective on both of them, and we were able to validate and characterize the affinity and the kinetics of 7 compounds for each protein, obtaining KD in the range from 3 to 8 micromolar for HMGA1, and from 1 to 32 micromolar for HMGA2. So quickly. And the advantages of this technique is the fact that it's a technique that allows you to follow the binding in real time. You don't need to level your sample. It's high throughput, and you need a very low amount of ligand. I forgot to tell you that when you immobilize your protein, you need around 10 microliter, 100 microliter of sample that is whose concentration is from 5 to 50 microgram per ml, so very low amounts. And this and the sensitivity of the instruments allows to get good signal for the interaction with small molecule and perform high throughput screening also on fragment libraries. It also is able to characterize the fastest transition and is very robust, so you can work on also with serum complex, blood complex sample or amplified sample. Quickly to recap, not only not all the system we studied were successful stories, but I guess that 83% of success is quite good. And the main problems are due to the stickiness of analyte. You cannot always solve this problem, even if there are tricks. And moreover, it's not always possible to switch configuration, especially if you are dealing with the interaction between a protein and a small molecule. But what's more important is the quality of the proteins you are working with because proteins that are made by people that do structural biology, so Marta, Paola, Stefano gives a beautiful signal. If you know everything about the stability, how you can treat your protein, you have a guarantee that your story will be successful. So just to close this part, when you put you try a setup for your experiment, you have always to keep in mind that failure is success in progress. So very briefly now, what's next? I told at the beginning that we are going to improve our facility that now is based on only 2 complementary techniques. We are going to improve this technique, upgrading our cryoptics delta. In this way, we will have 4 channels instead of 2, and so in this way we can increase the throughput and also decrease the amount of the library if we perform high throughput library screening. And also there is this new setup they developed that is the so-called fast kinetics that allows you to measure 6 times more sample at a time, because you don't have to inject increasing concentration of the analyte, but you inject the same concentration of the analyte for different times, and they developed a fitting strategies that allows you to get the same information. Then we also are going to upgrade our old micro calorimeter with a new one that allows an automatic robotic cleaning of the cells. And we are going also to upgrade our plate reader in order to be able to perform fluorescence anisotropy experiments to get the KD in a quite easy way, and as the instrument I'm let's say very I really looking forward to having my lab, because I tested at the time, is this Helix Plus that is based on a completely different system. It's based on DNA nano level, so the conjugation is completely different, and this can somehow solve problems of stickiness to the chip, and it gives somehow it is an high throughput technique that allows you to have overlapping from one hand information regarding wave, because you get kinetic and affinity information, but also you can characterize multiple binding sites that, as I told before, is not possible with the SPR and GCI. And it's very nice to see that you can follow also conformational changes of your protein on the chip. And I hope it was not too long, but I just wanted to thank my colleagues. First of all, Alberto, Letizia, that was a former colleague, my students, Martina, Lara, and Gabriele, Francesca, that last year joined us, and Luisa, that is our new entry and is working on a new system about the interaction between proteins and aptamers, and of course, all of you for your passion and attention.